Hello everyone, welcome back. In this video, we are going to study the question bank of Karnataka First PUC Biology Chapter 10, Cell Cycle and Cell Division. Here are first 1 mark question answers. Question number 1, what is karyokinesis? Answer, the division of nucleus is called karyokinesis. What is interphase? Answer, non-dividing phase of cell between the mitotic divisions is interphase. What is synapsis? Answer, the process of pairing of homologous chromosomes is called synapsis. What are chiasmata? Answer, the regions where segments of non-sister chromatids of the bivalents are exchanged are called chiasmata. Who first described mitosis in plants? Answer, Fleming. What are the two stages in cell cycle? Answer, interphase and M phase. Can there be mitosis without DNA replication in AS phase? No. Can there be DNA replication without cell division? Yes. What is the average cell cycle span for a mammalian cell? 24 hours. Between a prokaryote and a eukaryote, which cell has a shorter cell division time? Answer, prokaryote. Which of the phases of cell cycle is of longer duration? Interphase. Name the stain commonly used for color chromosomes. Answer, ornithine acetocarmine. Which tissue of animals and plants exhibits meiosis? Answer, animals, gonads, testes, spermatocytes, ovary, oocytes. At what stages of cell cycle does DNA synthesis takes place? Answer, S phase of interphase. Smaller lipid-soluble molecules diffuse faster through cell membrane, but the movement of hydrophilic substances is facilitated by certain transporters which are chemically. Answer, proteins. 2 marks question answers. Write the significance of mitosis or mitosis. Answer, significance of mitosis, distribution of equal number of chromosomes to the daughter cells which maintains a constant chromosome number. Mitosis increases the number of cells and hence so it contributes to growth. Significance of meiosis. Meiosis brings genetic crossing over and random distribution of paternal and maternal chromosomes to daughter cells. Recombination produces variations and variations are the source of organic evolution. Write any two differences between mitosis and meiosis. Answer, mitosis consists of single nuclear division. Meiosis consists of two nuclear division. In mitosis, crossing over is absent. In meiosis, crossing over is present. Distinguish cytokinesis from karyokinesis. Answer, cytokinesis is the division of cytoplasm and karyokinesis is the division of nucleus. What is G-quiescent phase of cell cycle? Answer, it is the cells that do not need no divide from there exist GO phase of the cell division phase to enter an inactive phase called GO. They remain metabolically active but do not proliferate, that is multiply phase where cell exists from cell division of it undertakes growth phase. Why is mitosis called equational division? Answer, the number of chromosomes in the parent and daughter cells are the same in mitosis, hence it is called equational division. How does cytokinesis in plant cell differ from that in animal cell? Here is the diagram for that. Telophase and cytokinesis in animal phase and plant cell. A is the animal cell and B is the plant cell diagram. Find examples where the 4 daughter cells from meiosis are equal in size and where they are found unequal in size. Answer, equal daughter cells, sporangia in plants, spermatid formation, unequal daughter cells, ovum and polar cells during oogenesis. What is the significance of meiosis? Answer: meiosis maintains the chromosome number constant in sexually reproducing organism. It is essential since the chromosome number is doubled after fertilization. It results in crossing over and recombination of genes. This brings in variation in the population. Meiosis in reproductive organs result in the formation of gametes. Analyze the events during every stage of cell cycle and notice how the following two parameters change. Number of chromosomes n per cell, amount of DNA content C per cell. Answer: G1 phase, DNA and chromosome content do not change. S phase, amount of DNA is twice that of the original cell. M phase, chromosome number per cell remains the same after M phase. DNA content is same as the original cell. Label the diagram and also determine the stage at which this structure is visible. Here is a diagram. Next, 3-mark question of cell cycle and cell division. Question number 1. Describe the events taking place during interphase. Answer: interphase, L interbetween BR phases aspect. The period between two mitotic divisions is called interphase. G1 phase, it is the most mitotic phase. It is the post-mitotic phase and takes place at the end of cell division. During this, active synthesis of RNA and protein takes place. S phase, it is the synthesis phase. During this phase, duplication of DNA and centriole takes place. G2 phase, it is the pre-mitotic gap phase. The synthesis of RNA and protein continues in this phase. Next, name the stage of the cell cycle at which each one of the following events occur. Chromosomes are moved to spindle equator, centromere splits and chromatid separates, pairing between homologous chromosomes takes place. Answer: first answer is metaphase, second answer is anaphase, third answer is zygote. Next, distinguish anaphase of mitosis from anaphase one of meiosis. Answer: anaphase of mitosis. Anaphase is the stage during which the centromere splits and the chromatids separate. The chromosomes move apart towards the opposite poles. These chromosomes are genetically identical. Anaphase 1 of meiosis. During anaphase 1, the homologous chromosomes separate while the chromatids remain attached at their centromeres. Hence, in anaphase 1, the chromosomes of each bivalent pair separates while the sister chromatids remain together. The cleavage of centromere is taken place in anaphase of mitosis, where it does not occur in anaphase 1 of meiosis. Next, 5-mark question answers. Question number 1. Explain with a neat label diagram the process of meiosis. And here is the diagram of interphase, leptotene, zygote, pachytene, diplotin, dikinesis, metaphase 1, anaphase 1, prophase 2, metaphase 2, anaphase 2, telophase 2. All the diagrams are here, that is the stages of meiosis. Next question. Describe the following: synapses, bivalent and chiasmata. Next, draw a diagram to illustrate your answer. Answer is: meiosis is a division which occurs in a diploid cell and gives rise to 4 haploid cells or nuclei. Therefore, meiosis is known as reductional division. Mitotic division takes place in 2 successive stages, namely meiosis 1 and meiosis 2. And here is the diagram of the both. Next is the textbook question answers. Cell division is an intrinsic property of a cell. Walter Fleming, 1882, studied the detail of somatic cell division and coined the term mitosis for it. There are 3 types of cell division: amitosis or direct cell division, mitosis or indirect cell division, meiosis or reduction division. Cell cycle. The sequence of events that occur between the formation of a cell and its division into daughter cells is called a cell cycle. It consists of two stages, a non-living growing interphase or interphase and a short dividing mitotic or M phase. Number 1. Interphase. The period between two mitotic divisions is called interphase. G1 phase. It is the post-mitotic division and takes place at the end of cell division. During this, active synthesis of RNA and protein takes place. S phase. It is the synthesis phase. During this phase, duplication of DNA and centriole takes place. G2 phase. It is the pre-mitotic gap phase. The synthesis of RNA and protein continues in this phase. Next, mitotic division. Mitotic division has two processes occurring simultaneously. They are karyokinesis and cytokinesis. Karyokinesis is a continuous process and for convenience divided into 4 stages. They are prophase, metaphase, anaphase and telophase. Prophase. It follows the interphase and characterized by the following features. The already duplicated chromosomes becomes progressively shorter and thicker due to condensation. The spindle fibers appears between the centrioles present at the poles. The nucleolus diminishes in size and finally disappears. The nuclear envelope is partly disintegrated. Next is metaphase. It is characterized by the following features. Spindle fiber are completely formed. The spindle fibers that are attached to chromosomes are called chromosomal fibers. The chromosomes aggregate in the center of the cell. They come to lie in the equatorial places to form an equatorial plate. And here is the diagram for that. Next is anaphase. Anaphase is characterized by the following features. The two chromatids of each chromosome separate completely to become daughter chromosomes. The two daughter chromosomes move away from the equator towards the opposite poles. During anaphasic movement of chromosomes, the centromeres lead the path and the arms trail behind. As a result, the anaphasic chromosomes appear V, L, J and I-shaped. Next, last phase is telophase. It is characterized by the following features. Here, reversal of the prophasic events occur. The daughter chromosomes moves and reach the opposite poles where they become thin and thread-like again. These threads overlap one another to form a fine chromatin network. The spindle fibers disintegrate and disappears. Reconstitution of a new nuclear envelope occurs. Nucleolus reappears. Next, cytokinesis. The division of the cytoplasm is called cytokinesis. A furrow develops in the middle of the cell. They deepen centripetally till the cytoplasm is divided into two equal parts. It forms two daughter cells. Next, significance of mitosis. The somatic chromosome number 2n in all the cells of an organism is effectively maintained by mitosis. The duplication of chromosomes occurs during the interphase and their orderly behavior during the different stages of mitosis maintain a constant chromosome numbers. The daughter cells produced as a result of mitosis are quantitatively and qualitatively identical. Mitosis of somatic cells helps in growth and development of multicellular organisms. It forms a means of multiplication in some of the tower organism, example amoeba, euglena, etc. Mitosis produces new cells for the healing of wounds and for regeneration. Next is meiosis. Meiosis is a division which occurs in a diploid cell or gives rise to 4 haploid cell or nuclei. Therefore, meiosis is also known as reductional division. Mitotic division takes place in 2 successive stages, that is meiosis 1 and meiosis 2. And here are the diagrams for that. Each of these show karyokinesis and cytokinesis. These 2 steps involved in the following. Here is the differentiation, meiosis 1. During this process, the diploid parent cell divides into 2 daughter cells which are haploid, hence it is a reduction division. Karyokinesis, prophase 1, it lasts for quite a long period and is studied under 5 substages. Leptotene, zygote, Pachytene, diplotin, diakinesis. Leptotin. The chromosomes are thin, long, and uncoiled. Each is a double chromosome consisting of two chromatids. These chromatids are held firmly. Each chromosome appears as a string of beads. The beads are the chromomeres. Zygotin. During this stage, pairing of homologous chromosomes, half of them maternal and half of them paternal. This pairing is called synapsis. The pairs so formed are called bivalent. Each bivalent consists of four chromatids and is therefore called a tetrad. The two chromatids of the same chromosomes are called sister chromatids, and the belonging to two different chromosomes of a homologous pair are termed as non-sister chromatids. Pachytene. Crossing over takes place by breakage and reunion of chromatid segment. After crossing over, the two chromatids of a chromosome become dissimilar. The points of interchange are X-shaped and are called chiasmata. Diplotin. Repulsion between homologous chromosomes begin. Each tetrad now appear in different shape, that is X-shaped, H-shaped or O-shaped. Repulsion results in terminalization of chiasmata, sliding of chiasmata towards the end of chromosome. Diakinesis. The nucleolus breaks down and disappear. Spindle fiber appear. Nuclear membrane breaks down. Chromosomes are released into the cytoplasm. Metaphase One. It is characterized by the completion of spindle formation. In animal cells, asters are formed around the centrioles at two poles. The paired chromosomes arrange themselves on the equator of the spindle fiber. Anaphase One. Here the spindle fiber contract, and this makes the bivalents move towards the opposite poles of the spindle without the division of centromere. The separated chromosomes or univalents are also called dyads because each of them consists of true chromatic which remains attached with each other at the centromere. Next, telophase. Chromosomes undergo uncoiling. A nuclear envelope is formed around each group of chromosomes. This nuclei do not reappear now. Meiosis 2. In this division, the two chromatids of each chromosome separates from each other and go to separate daughter cells. As a result, the number of chromosomes remains the same as produced by meiosis 1. Therefore, meiosis is called equational division. Significance of meiosis. Meiosis maintains the chromosome number constant in sexually reproducing organism. It is essential since the chromosome number is doubled after fertilization. It results in crossing over and recombination of genes. This brings in variation in the population. Meiosis in reproductive organ result in the formation of gametes. So this is all about your question bank of 1 mark, 2 mark, 3 mark and 5 mark questions with the textbook answers. If you want the more lessons, subscribe to my channel and check the playlist of my channel. Thank you.